Okay, quick introduction to the cell cycle. Your body needs to replicate cells because cells get damaged all the time. Your skin cells are falling off. If you drink a hot cup of coffee, you're damaging the cells in your mouth. And in order to do this, a cell needs to replicate DNA. So DNA is made of two strands, and these coil around each other, and that's called a double helix. Double because there's two, and a helix because it twists. Now, if I try to separate these two pipe cleaners by making a replication bubble and putting my finger in here and trying to pull them apart, you'll see it's a struggle. Replicating DNA takes a lot of energy, okay? That's what I want you to remember. Replicating DNA requires a lot of energy. Okay, so if we were going through this cell cycle, we need to take our chromosome complement, in this case a red chromosome and a yellow chromosome. We need to replicate the DNA. So these are centromeres, replicated chromosomes, and we need to undergo a process of mitosis in order to divide these up and to create two identical daughter cells. That's the process of mitosis. Okay, first of all I'm going to talk about the cell cycle. Okay, the cell cycle is a process by which a cell doubles, and to do this there are several different phases. So I'm going to start with gap phase 1, and at the end of gap phase 1 there is what's called a checkpoint, like a roadblock. DNA replication takes energy, and there's no point in doing it if your DNA is damaged. So the G1 checkpoint, gap 1 checkpoint, checks for DNA damage, and it checks that the environment is favourable. Replication takes a lot of energy, doesn't happen unless the environment is favourable. If the cell meets these criteria, it then goes forward to synthesis phase, S phase. S phase is followed by another gap phase, gap 2, G2, and it won't surprise you to learn there's another checkpoint. And at this checkpoint, the G2 checkpoint, the cell checks that the DNA is properly replicated, and it checks that the environment is favourable. That's the G2 checkpoint. If these conditions have been met, the cell then enters mitosis. During mitosis, there is a third checkpoint called the spindle checkpoint. This looks for unattached chromosomes. A cell with too many or too few chromosomes is bad news, and so the cell checks that all the chromosomes have been properly attached before proceeding. This is the cell cycle, and you need to remember there's 3 checkpoints: G1, G2 and the spindle checkpoint. Okay, so if we've met those conditions and the cell is about to enter mitosis, there are several phases of mitosis: interphase, prophase, metaphase, anaphase and telophase. Okay, if you looked under the microscope during interphase, you wouldn't see much. But during prophase, the chromosomes condense and become visible. So what we're going to do We're going to start with a parent cell with a yellow and a red chromosome. We're going to want to make two identical daughter cells. So the DNA needs to replicate. The centromere here in the middle is a massive complex of several hundred different proteins. Spindle fibres attach and help to pull the chromosomes apart. So, prophase, they pair. Metaphase, they meet in the middle. Anaphase, they pull apart. Telophase, you have two new cells. Cell 1 here and cell 2. So one more time. Prophase, they pair. Metaphase, they meet in the middle. Anaphase, they pull apart. And in telophase, you have two new cells. And that is the process of mitosis. Mitosis creates toes. Toes are part of your body. They are somatic cells, body cells. The purpose of mitosis is to produce two identical cells. They're diploid, and you must not confuse mitosis with meiosis. Meiosis produces eggs, gametes, okay, sperm and eggs. These are sex cells. They are genetically different because of independent assortment, crossing over. Unless you're a twin, you don't look like your brothers and sisters. And these cells are haploid. Okay, so when I was discussing the cell cycle right now, I was discussing mitosis.